Hi everybody, I am Dr. D.S. Thore. Now I share the script of today's lecture in front of you. Please wait. Welcome to the third lecture of course number PATH-365. Title of this course, Diseases of Field and Horticultural Crops and Their Management Second. Okay, so in today's lecture, we discuss about the powdery mildew disease seen in field and vegetable crops. Okay, so this is the most important disease seen in every field crops and vegetable crops. Okay, so this is the powdery mildew disease seen in wheat crop, pea crop, linseed, cucurbits, and corianders. Okay, respectively these causal organisms. For example, powdery mildew of wheat caused by Ericipi graminis variety Tritici, powdery mildew of pea caused by Ericipi polygoni, powdery mildew of linseed caused by Odium lini or Podospira lini, powdery mildew of cucurbits caused by Ericipi ciceroserum and Spirotheca filiginea. So this powdery mildew disease seen in cucurbits caused by two pathogens, one that is Ericipi ciceroserum and another pathogen that is a Spirotheca filiginea, and last one powdery mildew of coriander caused by Ericipi polygoni. Means this wheat, pea, linseed. This is the field crops, and this cucurbits, that is the horticultural crops, means vegetable crops, and coriander is also vegetable crops. Okay, so these different crops suffer the pathogen and produces the powdery mildew disease caused due to the Ericipi and these are Podospira pathogens. Okay, so just to see one by one, that is the first one, powdery mildew of wheat. Okay, so this powdery mildew pathogen is a obligate parasites. Obligate parasites means these pathogens or parasites always depends upon the other parasites. Means this pathogen is survival with the help of the other host or parasites. They cannot live alone. Okay, so this is the obligate parasites. So this obligate parasite infect to the wheat crops and produces the symptoms mainly on the leaf, younger shoots, inflorescence, and other younger tissues of the wheat crops. Okay, means initially small whitish spots seen on the leaf surface of the wheat. So these small means small whitish spots just grows on this infected leaf. Okay, means these pathogens appears superficial on the upper surface of the wheat leaf. Okay, so under development of the pathogens or under favorable conditions, this fungus, that is a whitish mass, spread to this entire leaf. So in this way, number of the leaf get infected by the fungus and showing the whitish mass. Means superficial flocculent whitish mats seen on the infected leaf of the wheat plants. Okay, so later on these pathogens or this whitish mass spread to the leaf sheath and floral bracts. Okay, so you this see this floral bracts or this panicle of these wheat plants. Okay, means these whitish spots also be seen on the leaf sheath as well as the floral bract. Okay, means this floral bract having the whitish mass. So these pods develop the seeds, and these seeds may be small in size because already this whitish mass seen on this inflorescence or these floral bracts. So that's why these uh, seeds develop from this infected floral bract or inflorescence. So this grain is always small in size and generally dried and maybe silvered. Okay, means these pathogens develop the symptoms on the leaf. Means in the most favorable condition, this whitish mass spread all over the parts of the wheat plant. So that's why retard the photosynthesis process, because this mass is coated entire or wholly surface of the leaf. So that's why retard the photosynthesis process. Okay, suppose this photosynthesis process is retarded in particularly wheat plants. So this formation of the airs from these infected plants, so this air is always small in size, having the only few grains are developed, and this grain may be discolored, silvered and dried. Okay, so in this way the infected wheat plants is totally weak, and these infected plants develop the ears. So this ears is also small in size or arrested these airheads. Okay, so this is the majorly symptoms seen on the leaf sheath, inflorescence or floral bracts, and also the ears. So this is the symptoms develop on the wheat plant caused by Irrecipe graminis variety TTC. Okay. Then etiological parts. Already I told these parasite is an obligate parasite. So this obligate parasite produces these symptoms on the infected leaf or any other parts of the wheat plants. So this pathogen develop the whitish superficial mats seen on the surface of the leaf or maybe other parts of the plants. Means this mycelia is a ectophytic mycelia or ectophytic type of mycelia. Ectophytic means this mycelia grows on the outer surface of the host cells, not seen on the inner side of the host tissue. Okay, ectophytic means this mycelia grows on the outside the host cells, means just a superficial surface of the host cells. And this ectophytic mycelia, uh, suppose this is the already this infected leaf having the whitish mass are seen. Okay, so this whitish mass, okay, means these ectophytic mycelia develop the spores, and these spores means this Irrecipe graminis variety TTC, this pathogen developed by sexually as well as the asexually reproduction methods. Okay, means this ectophytic mycelia grows into the host cells by forming the Hysteria. So finally develop the conidiophores. Okay, means this conidiophore, suppose this is the conidiophore arrives from this infected host tissue. Means this conidiophore generally develop the number of the conidia. Okay, and this conidia is formed in chain patterns. Means single conidium having the 10 to 12 conidia is produced, and this conidia is oval or maybe barrel shaped, haline in nature, and having the just single cells are produced, or uninucleated haline single spores is developed on the tip of this conidiophore. Okay, means this fungus reproduced by asexually methods. So finally this conidia is produced, and this conidia acts as a secondary source of infections. Okay, so before that, this fungus, this ectophytic fungus is also reproduced by sexually methods. Okay, by forming the sexual spore fruits that is known as the Clustothecia. So this is the Clustothecia. Clustothecia means closed ascocarp without opening, and this Clustothecia is a sexual spore fruit. Spore fruits means spores and spore bearing hyphae just gathered in a specific structure that is known as the spore fruits. Okay, so in this case, this Clustothecia is developed. Okay, so after germination of this Clustothecia, so number of the ascus and asci is produced inside this Clustothecia. Okay, means asci generally arranged or scattered irregularly on this Clustothecia. So this asci containing the 8 ascospores are produced. Means this type of the ascospores is produced in a single asci, and this asci generally produced within this Clustothecia. Okay, so these ascospores acts as a primary source of infection to these healthy one wheat plants. Okay, these ascospores means infected plant part debris in the soil, or ascospores present in these infected plants, and this infected plant part debris in the soil. So finally initiate the infection in the new healthy one plants. Okay, so keep in your mind that is the primary source of infection and secondary source of infection. That is a primary source means infected plant part debris in the soil, or Clustothecia bodies remain present in soil having the ascospores. This is the primary source, and secondary source, that is airborne conidia disseminated from these infected plants to these healthy one plants. Okay, means disseminate this conidia through wind or air current and directly deposit or landing on this healthy one wheat plant. So this healthy one plant is also infected. Okay, so this is the disease cycle perpetuation about this Ericipi graminis variety 3TC. Okay, then control measures, that is wider hose spacing, then reduce the irrigations. So these two cultural practices mainly adopted by the farmers to avoid this powdery mildew infection on the wheat plants. Then collect and destroy these infected plant parts just seen on these fields. So to control the primary source of infection. Suppose this powdery mass seen on the any plant parts of the wheat, so immediately you can apply the fungicide, that is Carathine 0.05% or Bavistin 0.1%, Vetable powder 0.3%, and Benlate 0.1%. So this is the number of the fungicides is used for spraying purpose over these wheat crops to against the powdery mildew pathogens seen on the wheat plants. And just growing resistant variety, that is HD 2204 CPAN 1922. This is the two most important variety grown every farmer against these powdery mildew pathogens. Okay, so this is the information about powdery mildew of wheat plants. Okay, then this is the second most important crops also in this powdery mildew disease. Okay, so you see these pea plants having the powdery mass are seen. Okay, means majority these pea plants suffer to this irrecipe polygony fungus at the time of the flowering and also pod development stage. Means at the time of the flowering, the whitish powdery growth seen on the leaf surface, seen on the pod surface, or seen on the uh, flower also. Okay, means initially just the whitish dots or spots seen on the leaf portion or pod portion or maybe flower portions. Okay, so under wet conditions or under humid condition, this powdery mass is just coated on the both side of the leaf or maybe pod surface or flower portions. Okay, means this powdery mass spread to the entire surface of the leaf or pod surface or flower portion of these pea plants. Okay, means as the plant become old, the color of this powdery mass change to just brown in color. So you just see this brown color. This is the white color and brown color. May this brown color represent as a formation of the Clustothecia bodies develop on these infected lesions. Okay, means this is the indication for development of the Clustothecia bodies. And this white color represent as a formation of the conidia. Means white colors develop the conidia by asexual method, and this brown color represent as a formation of the Clustothecia having the ascospores are produced. Okay, so in the favorable condition, this whitish mass spread to entire surface of the leaf. So in this case, also retard the photosynthesis process, and this plant growth is affected adversely. Okay, suppose this whitish mass seen on the pod surface. Okay, so these pods having the seed, so these seeds may be shriveled and discolored. So this infection also be seen when these pathogens seen or infect to these pods and showing the whitish mass. Okay, so this is the major symptom seen on the leaf, also means leaf, stem, then pods and also flowers of the pea plants. Okay, then causing pathogen, that is irrecipe polygon. Okay, so already I discussed with you regarding the irrecipe characters or irrecipe genera or etological parts of this irrecipe. Means this irrecipe reproduced by two methods. One is the sexual, means finally develop the conidia, and ascospores reproduced by sexually method by forming the Clustothecia. Okay, means primary infection generally takes place through Clustothecia bodies present in soil are present in the infected plant part debris in soils. And secondary source of infection generally takes place through this conidia by means of wind or air current. Okay, so this is the disease cycle of these irrecipe polygony. Means this is the infected plants having the whitish mass are seen, and this whitish mass turns to the brown in color. Means already Kustothecia bodies is developed. So this is the Kustothecia having the one characters to properly identify the irrecipe genera. So on the surface of the Clostothecia fruiting bodies, the mycelioid type of the appendages are seen, mean just the thread-like structures. Okay, so this mycelioid type of the appendages seen on the surface of the Clostothecia. This is the indication or mainly characters of the irrecipe genera. Okay, so this Clostothecia having the ascus generally produced. So this is the ascus having the 8 ascospores are produced. Means ascospores generally produced inside this ascus, and this ascospore directly deposit or landing on the healthy one host tissue. So these healthy one host tissues become infected by these ascospores by forming the hosteria. Okay, means this hosteria suck the cell sap or infect to this healthy one plants. Means this healthy one plant become infect and showing the whitish mass. So this is the disease cycle of the irrecipe polygony. Okay, then control measures, that is the sanitation programs, means infected plant parts should be collected and burned properly. Then use the disease free seeds, because this powdery mass also be seen on the surface of the pods, and these infected pods having the seeds, so these seeds may be contaminated, so avoid these infections. So this is before the sowing of the seed, so these seeds generally use disease-free plots or plants. Then early sowing of the crop gives it sufficient time for growth and reproduction before the actual disease is set. Okay, means to avoid the disease by adopting the early sowing time. Okay, then crop rotation with sugarcane, cereals crops, gram crops, pigeon pea, because this powdery mildew disease cannot be seen on the sugarcane or gram or pigeon pea crops. So that's why these pea crops crop rotate with the other non-host crop. Then growing resistant variety, then spraying the fungicide over this pea plant, that is a sulfax or 80% vegetable sulfur at the rate of 3 kg per hectare. So just take the 3 kg vegetable sulfur, dissolve in 600 to 800 liters of water and make the final volume. And this final fungicide volume is sufficient for spraying 1 hectare area of the pea fields. Okay, means 2 to 3 subsequently spraying should be necessary to avoid this powdery mildew disease in the pea at 10 to 15 days interval. Okay, then application of the vermicompost to inhibit the spore germination and development of the fungus in the field at very low concentrations. Okay, so this is the vermicompost just applied in soil to inhibit the uh, production of the Clostothecia body in soil. Okay, then powdery mildew disease of the linseed. So this is the linseed plants and having the powdery mass are seen. Okay, means this disease, this disease means powdery mildew disease seen at the time of the flowering or at the time of the capsule formation or seed setting in the linseed plants. Okay, means a powdery mass just seen on the leaf portion, stem portion, capsule portion, or on the surface of the seed. So rapidly spread this whitish mass to cover to the entire surface of the leaf or any parts of the plants. Okay, means severely infected leaf just dries, withers, and defoliated from these linseed plants. Okay, means defoliated the leaf due to the whitish mass seen on the leaf surface. Means retard the photosynthesis process. So these plants having the leaf become chlorated, and this yellowing leaf generally defoliated from these linseed plants. Okay, suppose this powdery mass seen on the capsule and seed setting time of the plants, so retard the or reduce the yield of particularly linseeds plants, and also retard the or reduce the quality of the seeds, because these seeds is chappy in appearance or maybe discolored in nature, or silvered these seeds produced from these infected linseed plants. Okay, so this is the symptoms produced from the linseed plants. Okay, caused by the odium linea or podospira linea. Okay, so this podospira linea fungus reproduced by asexually by forming the conidium. So this process is the same in Elicipi genera. But this Spodospira linea reproduced by sexually method by forming the perithecium spore fruits. Okay, in regarding the Elicipi, Pustothysia spore fruits are produced, but in Podospira, Perithecia spore fruits are produced. Perithecia generally flask-shaped spore fruits having the asci and ascospores are produced. Means number of the asci just produced inside this perithecium fruiting bodies, and this ascus having the near about 6 to 8 ascospores is produced from a single ascus. Means primary infection is always takes place through these ascospores present in the infected plant parts or perithecium fruiting body, just survival in the soil for a number of days. So this is the initial infection to produce the disease in the linseed plants. And secondary source, that is a conidia spread through irrigation. Sorry, this airborne or windborne. Means conidia disseminated from one plant to the another plant through wind agency or air agencies. Okay. Then control measures. This is the most common management practices used or adopt uh, by the number of the farmers to avoid the powdery mildew disease seen in the linseed, just the same as wheat plants or pea plants. Okay, so this is the, some cultural methods or cultural managements. Then some chemical managements is used for controlling this disease in plants. Okay, then cucurbits. So these cucurbits means cabbage, cauliflower, sorry sir, these melon crops and gourds crop means watermelon, muskmelon, then snake gourds, then cucumber. So these crops comes under the category of the cucurbits. Okay, so these cucurbits crops suffered and showing the whitish mass seen on the parts of the plants. Okay, means initially small whitish dots or whitish spots seen on the surface of the leaf or surface of the fruits or surface of the flowers, etc. Okay, means on the cucurbitaceous crops, initially this whitish mass seen on the leaf, majorly seen on the upper side of the leaf, showing the whitish superficial spots are seen, and these spots enlarges, forming the large patches or this whitish mass coated over this entire surface of the leaf or other parts of the plants. Means this whitish mass just spread from this leaf to the petiole portion or maybe stem portion or fruit portion. Okay, means this whitish mass turns to the black in colors. So this black color represent as a formation of the Custothecia. Okay, means in the severe infection, this infected leaf is defoliated from these infected vines of these cucurbitaceous crops. Okay, means retard the photosynthesis process. Okay, so this infection or this whitish mass also be seen on this development of the fruits. So these fruits is always small in size, or these fruits produce misshaped. Means these fruits is not properly shape is developed because already this mass is seen or fungus attack to this developing fruits. Means these fruits from affected plants are very small in size and misset or discolored fruits. So in this way, the considerable reduction in the yield is reported from this powdery mildew infection seen on the many cucurbitaceous crops. Okay, so this is the pumpkin plants or vines suffer this powdery mass. So this is the cucumber plants having the whitish mass seen on the leaf and also fruits. So this is the powdery mass seen on the crown portion or stem portion or just branches portion of these squash plants. So this is the powdery mass seen on the pumpkin fruits, okay, or these stalks of the fruits. Okay, means this powdery mass coated over these fruits of the pumpkin. Okay, then causal organism EDCP, Ciceroserum and Spirotheca fuligina. So this powdery mildew, this is seen in the cucurbitaceous crops by two pathogens. One is the EDCP and another is the Spirotheca. Already I discussed with you regarding the etiological parts of the EDCP. Okay, so this Spirotheca, so this Spirotheca fuligina pathogen also develop the conidia on the tip of the conidiophore, and this conidia is always a barrel shape having the proper size. Okay, so this Spirotheca pathogen reproduced by sexually method by forming the Pailthecia spore fruits. So inside the Pailthecium spore fruits, number of the asci generally produced, and this single asci having the 8 ascospores are produced. Okay, means same reproduction method, that is the sexual and asexual methods. Then primary infection is same infected plant part, debris in soil or Custothecia or and Pailthecia generally left over in the soils, and secondary source, that is conidia spread through wind or air currents. So this is the ascogenous hyphae and asci generally develop, and this is the conidiophore and conidia is seen. So this is the conidia, means this conidia form in chain pattern at the tip of this conidiophore. So this is the disease cycle of the EDCP and Spirotheca, means this is the EDCP genera by forming the Custothecia and by forming the ascospores. Okay, this is the asexual reproduction method by, means in this case the conidia is formed, and this is the sexual cycle. So this crustaceea generally develop. So inside this crustaceea, ascospores are produced. Means in sexual cycle, the ascospores produced by sexual methods. Okay, then control measure, that is the crop rotations at least 1 or 2 years without cucurbits is grown near to these cucumber plants or other grass crop or melon crops. Okay, then all the infected plant parts should be destroyed. Then use a tolerant or resistant variety of the cucurbits, then spraying with carbendazim, dinocap, thiophanate-methyl, benomyl, or recently this wettable sulfur is used to avoid the disease seen in the cucurbitaceous crops. Okay, then this is the last vegetable crops suffer to this powdery mildew pathogen and develop the symptoms on particularly coriander crops through EDCP polygony. Okay, means in this case this disease first appears as a minute discolored specks seen on the surface of the small sized leaf of the corianders. Okay, means this whitish mark just seen on this young leaf produced from this coriander crops. Okay, so under favorable condition, this mass seen uh, or spread over these whole plants of the corianders. So finally, this whitish mass turns to the brown in colors, means formation of the uh, crustaceal bodies. Okay, so this is the symptoms produced by these powdery mildew, that is ERCP polygony on the coriander crops. Suppose these coriander plants or leaf having the whitish mass and just prefer for eating purpose. So these farmers or any person cannot prefer such a infected coriander leaf or plants for eating purpose. Okay, then causal organism ELCP polygony already discussed these ecological parts of the ELCP polygony, and also perpetuation, that is a primary source and secondary source. So this is the asexual disease cycle of the ELCP polygony by forming the conidia on the tip of this conidia. Okay, means this conidia disseminated and just landing on this healthy one host tissue. So these spores germinate by forming the host area. So this in healthy one host tissue become infected, means already this whitish mass seen on the leaf of the corianders. Okay, so in this way, secondary source of infection or spread of disease from this infected plant to this healthy one plants. Then in case of the sexual cycle, okay, so these infected leaf or any parts of the corianders develop the brownish colors, means this whitish mass turns to the brown in colors, means finally develop the sexual spore fruits. So these sexual spore fruits formed by fusion of the female gamete and male gamete. Antheridium is a male gamete and Ostrogonium is a female gamete. So male nuclei migrates from this Antheridium towards the Ostrogonium. So finally male and female gamete fuse to each other and finally form the clostothesia. So this is the Clostothecia having the myceloid type of the appendages are present, and inside the Clostothecia ascus generally produce. So single ascus having the 8 ascospores are produced, and this ascospore liberated from this asci and just deposit on the healthy one host tissue. So this healthy one host tissue become infected. So in this way primary infection and secondary infection generally takes place or seen in coriander crops. And this is the management point. This is the same management points. Means use the balanced dose of the fertilizers, then early planting, then collection and destruction of the infected plant debris. So this is the cultural management practices adopt in cycle of these coriander crops, so you can control or eliminate the primary source of infection. And this is the fungicide application to these coriander crops, but mainly eating parts of the coriander, that is a leaf. Suppose you can heavy or increase the number of this fungicide sprays over these coriander crops. So these fungicide residues maybe remain present on this coriander leaf. So in case of the corianders, only one or two sprays should be done. Okay, that is use the sulfex carotene, then sultap, then carbon enzyme, thiophanate methyl. So this is the fungicides is also recommended to control the powdery mildew disease seen in the coriander, then growing the late and early maturing variety, means to avoid the disease seen on the coriander by adopting or growing the variety of the corianders is early maturing type of the variety. Okay, so this is the today's lectures. Okay, so in today's lecture we studied the powdery mildew disease. Okay, so keep in your mind about the information regarding the powdery mildew pathogens. Okay, means in case of the symptoms, the whitish mass just seen on the all the parts of the respective plants, means may be seen on the leaf, may be seen on the stem, may be seen on the pods or any reproductive organs. So these whitish mass spread to this entire surface of the plants. Okay, so in case of the severe infection, this whitish mass coated over the all the infected plants. So that's why retard the photosynthesis process. So retard the photosynthesis process in particularly crops. So this effect can be seen on the reproductive parts, or in case of the seed or in case of the pods. So these pods generally produced is only few number from these infected plants, or only few seeds are produced from single pods. Okay, means retard the quantity and quality of the seeds as well as the that pods. So these indications generally seen in the powdery mildew disease seen in the every crops. Okay, this is the symptom point. Then causal organism, that is Ericipe and Spirotheca. So these two genera is responsible to produce the powdery mildew disease seen in the respective crops. Okay, means this Ericipe pathogen reproduced by two methods. One is the sexual and another is the asexual. Suppose this pathogen reproduced by asexual method, so finally conidia is produced, and this conidia acts as a secondary source of infection. But in case of the sexual reproduction method, the Crustothecia sexual spore fruits are produced. Crustothecia, it is a sexual spore fruits. So inside the Crustothecia, number of the ascus are produced, and these ascus containing the ascospores are produced. And these ascus and ascospores liberated from this Crustothecia and directly deposit on the healthy one plant. So This is the primary source of infections. Okay, means primary infection, infected plant part debris in soil, infected seeds or maybe Clostridia debris in the soil. And secondary source, that is the airborne conidia. Okay, then in respect to these managements, so first management, that is a cultural management point, that is sanitation programs. Then second one, growing early maturing variety. Then third one, wider row spacing, means planting that seeds at the proper space. Okay, then growing a resistant variety and spraying the wettable sulfur or dinocap or carathine, these fungicides is also recommended to avoid the disease. So this is the, just a summary of these 2 days lectures. Okay, thank you.